Hey Bio Buddies, welcome to Energy Reactions in the Cell Part 4. I know, when is this ever going to end? Well, take heart, June is coming. It'll be here before you know it. Alright, this particular episode is going to be on cellular respiration and how it works. And when we look at the term respiration or hear the term respiration, a lot of folks think right away of breathing. Well, if you were to ask a typical second grader, why do you have to breathe? His or her answer would invariably be, because I need to live. You have to breathe to live. Well, all in well, but why do you have to breathe to live? If you have a smart second grader, they might turn around and tell you, hey, you need oxygen. Breathing gives your body oxygen. Cool. But the question still becomes, why do you need oxygen to live? So, that's what we're going to be investigating in this next section. All right, our learning objectives for this lesson are as follows. Much like our previous lesson on photosynthesis, you should be able to define the process of cellular respiration and supply its summary equation. You need to be able to distinguish between aerobic and anaerobic chemical reactions and break down the process of cellular respiration into its component parts. Therefore, you should be able to list both the reactants and products of glycolysis, pyruvate conversion, citric acid cycle, and electron transport. In addition, you should be able to describe each of these processes. Well, this is going to take some practice, so let's go ahead and get started. First off, cellular respiration, as defined by a biochemist, would be that process by which oxygen is used to help break down food and release energy. So going back to that second grade question, why do we need oxygen? Or why do we need oxygen to live? Well, we need that in a series of chemical reactions that allow us to break down food and release energy for each of our cells. If we look at the summary equation below, you'll probably notice that this is really similar to photosynthesis. As a matter of fact, if we take the summary equation for photosynthesis and turn it backwards, we've got cellular respiration. The products of photosynthesis, glucose and oxygen, are actually the reactants for cellular respiration. The products of cellular respiration, CO2, water, and energy, are going to be the reactants for photosynthesis. One of the chief differences here is we're not going to be talking about light energy, we're going to be talking about chemical energy. Chemical energy formed in the molecule ATP, of which you are familiar already. Now, before we get into describing the actual processes involved with cellular respiration, two important terms. Aerobic, sounds like air, right? Well, that refers to any reaction which requires oxygen. This gentleman here is performing an activity that requires oxygen and oxygen balance to keep him going. Its counterpart, anaerobic, means the process does not require oxygen directly. Now, this guy still needs oxygen down here, but he's relying a lot on stored ATP energy to actually go ahead and have one explosive effort. Certainly, if he was going to go ahead and maintain that effort, he'd still need oxygen. Nevertheless, we're going to see two different forms of chemical reactions involved here. Some of them are aerobic and some of them anaerobic. All right, now for the nitty-gritty. Before we look at the cellular respiration process in detail, we have to look at something called glycolysis. Glycolysis really isn't a part of cellular respiration, but it always has to happen before respiration takes place. So we're going to treat it as if it's part of cellular respiration anyhow. This occurs out in the cytoplasm. You don't need oxygen, so it's completely and totally anaerobic. All you need is the appropriate enzymes and a little bit of energy to get this started. So the requirements for these reactions, the reactants, we're going to go ahead and break apart a molecule of glucose. The term glycolysis literally means to break apart glucose. Glyco referring to glucose, lysis to break apart. In order to get this started, we need some activation energy, so we're going to go ahead and pump in 2 ATPs worth of energy. And we're going to need to strip away some hydrogens. To do that, we're going to use one of the hydrogen acceptors that we introduced in an earlier lesson, nicotinamide adenine dinucleotide. I'm just going to call it a NAD. So I'm not trying to say anything nasty. We'll just call it a NAD. What actually happens here? The 2 molecules of ATP are needed to go ahead and help break apart that 1 molecule of glucose. When we do this, we end up with 2 smaller molecules called pyruvate. Now these are 3-carbon compounds, but they're not exactly a half of a glucose. They've got a couple of hydrogens missing. So those hydrogens are released and have to be picked up by the 2 molecules of NAD to form NADH. Therefore, our products are going to be a couple of pyruvates, 2 NADHs, but in the process, this is really cool, we actually release more energy than we put into it. So while we invest 2 ATPs, we get 4 ATPs out of it. So we actually end up with a profit of 2 net ATPs. Let me show you this in a flowchart. Okay, we already know that our ultimate food molecule is glucose, C6H12O6. Well, our goal is to go ahead and break this apart, break the chemical bonds that hold glucose together, and release some energy. Well, in order to do this, you have to invest some energy. It doesn't happen by itself. How much energy? 2 ATPs worth of energy. When this happens, we're going to break apart glucose into 2 pyruvate molecules. Now, the pyruvate molecules, like we explained, are not exactly half of a glucose. Each of these are composed of 3 carbons, 5 hydrogens, and 3 oxygens. We're missing, and there's 2 of these, we're missing 2 hydrogens, 1 from each molecule. Well, the hydrogens are released here, and they got to be picked up by somebody because we're going to use them for an important process a little bit later. So we're going to have NAD swoop in, pick up our hydrogens, and exit as NADH. I can write an H. Now, what's really cool about this process is that during all of these reactions, we actually end up releasing some energy, releasing some stored energy in the chemical bonds of the glucose molecule. How much energy? Well, enough to go ahead and build 4 ATPs worth of energy. So in this first step, we're actually ahead of the game. We've actually made some energy. Kind of cool. Okay, here's that same flow chart, a little bit cleaned up. It's saying the exact same things. The reactants I've written in green, the products in blue so you can trace your way through it. I've left out some things here. For instance, when we say we're making ATP, it's not quite as magic as that. You know, you still have to have ADP plus a free phosphate being converted to ATP, but hopefully you get the idea. Now, Ideally, you want to be able to explain this process, not just in terms of products and reactants, but actually explain how the process works and how the process progresses. Now, you can do this with a flow chart, you can do this with a paragraph form. The only thing is, you do have to go ahead and explain your way through it. Do not simply memorize the flow chart as unconnected arrows and symbols. When you practice this, practice explaining your way through it, because that's what you're going to have to do on a test or a quiz. All right, we've broken apart a glucose molecule, and we got some energy out of it. That's the good news. Well, the bad news is that net profit of 2 ATPs worth of energy is not enough to keep a complex multicellular organism alive for very long. But the good news is that we can go ahead and harvest a whole lot more energy from what remains of that glucose molecule, which is now a couple of pyruvates. Well, the next set of reactions are called the Krebs cycle reactions, and they were originally discovered, kind of sort of worked out by a guy called Hans Krebs in the 1930s. They're also referred to as the citric acid cycle or the tricarboxylic acid cycle. You can call them any of those three and be perfectly fine. Now, it turns out that the first step of the Krebs cycle isn't quite so cyclic, so we're going to separate that out and call it pyruvate conversion. But in actuality, it is part of the Krebs cycle, but it's going to make it a whole lot easier to kind of chunk this out. So PC, pyruvate conversion, occurs within the mitochondria. So in order for this to happen, we're going to use our 2 pyruvate molecules that we just ended up with after glycolysis. We're going to break them apart a little bit more and strip off some hydrogens, so we're going to need NAD again. We're going to need a carrier molecule at the end of this to go ahead and carry it into the next set of the reactions, the true cycle portion. And that's a special molecule we'll call coenzyme A. So enzymatically, each pyruvate molecule is going to be transformed into something called acetyl coenzyme A. In this process, we're going to go ahead and strip off a carbon dioxide and 1 hydrogen for each molecule of pyruvate, and that's going to convert the pyruvate into an acetyl group. The acetyl group is then going to get picked up by coenzyme A, and it's going to be called acetyl-CoA, or acetyl coenzyme A. The hydrogen that was released is picked up by NAD to form NADH. Right? So we'll end up with a couple of NADHs. The carbon dioxide is some of the carbon dioxide that's a waste product of photosynthesis. And our pyruvate is going to be broken down and converted to acetyl coenzyme A. So again, let's look at this in flowchart form. All right, we're going to pick up where we left off. And where we left off at the end of glycolysis is you had 2 pyruvate molecules. And recall each pyruvate had 3 carbons, 5 hydrogens, and 3 oxygens from that original glucose molecule. Of course, there was 2 of them. What we're going to do is break this pyruvate down into something called an acetyl. Well, in order to get to an acetyl group, we have to go ahead and strip off from each pyruvate a CO2 molecule and a hydrogen. We're going to need that hydrogen later, so it's going to get picked up by NAD, and we're going to end up with NADH. And if we subtract a carbon from our original pyruvate, we end up, well, 1 carbon from 3 is 2. And we're going to subtract a hydrogen from our original pyruvate. So 1 minus 5 is 4. And we have 2 oxygens to deal with, so each of the pyruvates are going to lose 2 oxygens. They only had 3 to begin with, so they only have 1 left. Right? And of course, this happens to each pyruvate, so it happens twice, right? So we've got 2 CO2s being released, 2 hydrogens. You end up with 2 NADs. And of course, you end up with 2 acetyls. Okay, here's my acetyl group. Well, the acetyl is not going to be able to go ahead and participate in the next set of reactions unless it's carried into the next set of reactions by a little helper. And our little helper molecule here is going to be referred to as coenzyme A. And coenzyme A doesn't really change the acetyl group in any formative way. It just acts to carry it to the next set of reactions. So we're going to end up with the same amount of carbons, hydrogens, and oxygens carried by the coenzyme A. And thus we call this product acetyl coenzyme A. And, just as before, this happens twice, so we got 2 of these and 2 of those. Okay. So here's that flow chart, a whole lot neater than it was. Again, that which is in green is going to be the reactants and the products in blue. So we start out with our 2 pyruvates. We're going to go ahead and strip off our CO2 and hydrogens. Hydrogens get picked up by NADs, and we're left with an acetyl group. If you do the math, we have the appropriate remaining numbers of carbons, hydrogens, and oxygens. And then acetyl coenzyme A is going to swoop in, help carry this acetyl group into the next set of reactions. Again, practice this on your own, and you can combine it with the previous step, glycolysis. But as always, make sure that you're able to explain your way through it. The arrows are there for a purpose. They're showing you what's happening, what are reactants, what are products. Be able to explain your way through it. All right, these next set of reactions will show the complete destruction of what's left of our glucose molecule in the form of that little acetyl group. And this happens in a cyclic series of reactions, which is truly the Krebs cycle. Now, all this is going to happen in the mitochondria. We're going to use that acetyl group carried by coenzyme A and break it apart all the way. To do that, we're going to have to go ahead and strip off hydrogens, some of which are going to get carried away by NADs, and some of which are going to get carried away by FADH. We're also going to go ahead and strip off the remaining carbons. Now, to do this, we have to go ahead and strip it off in the form of carbon dioxide. Problem is that our acetyl group no longer has enough oxygen to even form carbon dioxide. So we're going to have to use some oxygen from another source. I kind of sort of show it here as an aerobic process, and in reality that's not strictly true, because the oxygen here is harvested from water molecules. So we don't really have to input pure water or pure oxygen. However, if you want to kind of sort of see what's going on molecule by molecule, it's easier to see it if you just trace the oxygen. So you'll just have to trust me on this. When acetyl coenzyme A enters the Krebs cycle, the coenzyme A is removed, and that acetyl fragment is going to be completely broken down. Again, the hydrogen is going to get stripped off and picked up by either NADH or FADH2. The remaining carbons are going to be used as a waste in CO2, and we actually get a little bit of extra energy out of this. Some more ATPs. Still not enough to keep us alive, but at least it's something. So let me show you the short magical version of this process. All right, so we're going to go ahead and give you a summary of the citric acid cycle here. The summary, because if we gave you the full story now, we'd be here for a while. Secondly, if we gave you that full story, we'd be going through a whole lot of detail that you probably don't really need to know to understand the basics of what's going on. So let's leave it as simple as possible. All right, so where we left off with the pyruvate conversion step, we had two acetyl coenzyme A molecules. And don't worry about the coenzyme A for the time being. That just acts as a carrier. Remember, the acetyls are composed of 2 carbons, 4 hydrogens, and an oxygen from the original glucose molecule. And of course, there's 2 of those. So everything's multiplied by 2 here. Anyway, our goal here in the rest of the citric acid cycle is to go ahead and destroy this molecule completely. All of the hydrogens are going to be carried away and used later. The carbon and the oxygen have to be removed as CO2. Problem is, we don't have enough oxygen to make a CO2. So we're going to have to go ahead and add some stuff. Here's how it works. Once again, we're going to take those acetyl coenzyme A's and we're going to go ahead and strip off the coenzyme A. So what's left is just those acetyl fragments. Again, 2 carbons, 4 hydrogens, and 1 oxygen from the original glucose. But remember, there's 2 of these, right? So we actually have 4 carbons to deal with, 8 hydrogens, and 2 oxygens. Well, if we want to go ahead and strip our carbons and oxygens off as CO2, we do indeed have 4 carbons there, but we certainly don't have 8 oxygens. So we have to go ahead and borrow oxygen from another source. So we're going to use 3 oxygens and pump them in here. That'll give us enough to have enough oxygen to make CO2. In addition, we're going to have to strip off all of these hydrogens. Well, 2 times H4, that's 8 hydrogens. 6 of these are going to be handled by NAD to make 6 NADHs down here. And the other 2 are going to be handled by FADH to make FADH2. At the end, this entire molecule will be destroyed, and we end up with a kind of a bonus. As that molecule is destroyed, we're going to go ahead and release enough energy held in the chemical bonds to make 2 more ATPs worth of energy. Good, in that we make more energy out of it. Bad news is that that's still not enough to go ahead and keep a complex multicellular organism alive. Bottom line, however, we're going to go ahead and harvest even more energy in the next set of reactions. And that's where all the action happens in terms of making lots and lots and lots of ATP. Okay, our last series of reactions also occurs in the mitochondria, and we're going to use all of those hydrogens and all of the high energy electrons from those hydrogens that we kind of sort of harvested from glycolysis, pyruvate conversion, and Krebs cycle. So in the process, we had 2 NADHs from glycolysis, 2 from pyruvate conversion, and 6 from the Krebs cycle, giving us a total of 10. We also ended up with 2 FADH2s from the Krebs cycle. That accounts for all 12 hydrogens that were once attached to a glucose molecule. And we're going to get all the energy that is associated with those hydrogens to make lots and lots of ATPs. Well, the hydrogens have got to go somewhere at the end, so we're going to need some oxygen to go ahead and pick up those hydrogens and carry them out as waste in the form of water. So, in essence, energy associated with those electrons and hydrogens are going to be used to make ATP. Oxygen acts as our final hydrogen acceptor to go ahead and form water and carry those hydrogens out as a waste. We're left off with NADs, which can be recycled in glycolysis, pyruvate conversion, and Krebs. FADs, FADHs, which can be used and recycled back in the Krebs cycle. Water is going to be our waste, and hey, that's a lot of energy. 26 to 28 ATPs is certainly enough to go ahead and keep complex multicellular organisms alive. So, let's look at how this actually happens. As in Krebs cycle, electron transport is actually a complex series of reactions that involve a bunch of steps. We're going to keep it as simple as possible and just give you the summary. All right? So electron transport implies that we're going to get energy from electrons, but remember the energy from the electrons is actually going to be carried by those hydrogen carriers, which are NADH and FADH respectively, or FADH2. And remember, in glycolysis, we ended up with 2 NADHs. We said they were going to be important later. With pyruvate conversion, we ended up with a total of 2 NADHs. And in the rest of the Krebs cycle, we ended up with 6. So we have a grand total of 10 NADHs to be fed into the system. The Krebs cycle gave us 2 FADHs, and 10 plus 2 gives us 12 hydrogens, which is exactly what we had attached to the original glucose molecule. So the quickest summary we can give you here. We're going to feed in our 10 NADHs. We're going to strip off the hydrogen and use their attendant electrons to go ahead and help convert ADP into ATP. And we're going to end up with NADH. We're going to take the 2 FADH2s from Krebs cycle. We're going to strip off the hydrogen, use the energy from the attendant electrons to make a whole lot more ATPs or convert ADPs into ATPs. The FADH, remaining FADH, can be, again, recycled and reused. The hydrogens that were stripped off are going to combine with oxygen. And the oxygen here is just used to combine with the hydrogen to make water, and that's going to be our waste product. We should have 6 waters as a result. And coincidentally, or not coincidentally, we should have 6 waters as a waste product from respiration. Now, what seems like magic here is the fact that we get all these ATPs. Now, I'll show you how those are generated here in just a sec. But before we go too far, why is it 26 to 28 ATPs? Why don't I just give you a proper number here? Well, here's where it becomes a little bit tricky. Turns out that for every FADH that you pump into the system, you could actually get an average of 1.5 ATPs out of that? Well, we had 2 FADHs, so that gives us 3 ATPs from our FADHs, or FADH2s. For every NADH that you pump into the system, you can get an average of 2.5 ATPs. Well, if we had 10 of these, that's 25 ATPs. 25 plus 3 is, hey, 28. So, yeah, it's possible to get 28 ATPs out of this. But recall that of the 10 NADHs that we had, 2 of them came from glycolysis. And 2 of them came out in the cytoplasm. And it turns out that some cells can pull those NADHs right back into the mitochondria and into electron transport without the use of energy. But some cells can't. Some cells have to go ahead and use a little bit of energy for active transport. And it will take them 1 ATP per NADH to pull it across the membrane. Well, since there's 2 of those outside the membrane, that would go ahead and subtract 2 ATPs from our total and give us 26. So, yeah, the numbers are either 26 or 28, depending upon the cell. But hey, think of it. Grand totals in glycolysis, you had 2 ATPs. In Krebs cycle, you're going to add up 2 more. In here, you're going to have either 26 or 28 ATPs. So bottom line, you're going to end up with anywhere from 30 to 32 ATPs as our grand total. Not bad. But we need to go ahead and look at how we generate the ATPs here, because, yeah, it just seems like a magic box here. So let me take some of the magic out for you and show you how this thing actually happens. Right, this wonderful diagram is meant to show the inner workings of a mitochondrial membrane. Here's our membrane with our phospholipid layer and some proteins embedded in, right? And the protein, there are various protein complexes that are designed to strip off hydrogen from either NADH or FADH2, converting these back into NADs, which could be used and reused over again, and FADHs, leaving a bunch of hydrogen ions on this side of the membrane. And this is kind of cool because we have another enzyme, another protein, rather, embedded in the mitochondrial membrane, which is a special channel protein allowing hydrogen ions to flow from point A, areas of high concentration, to point B, areas of lower concentration. Sounds a little bit like facilitated diffusion, because it is. Well, here we have a flow of charged particles going from high concentration to low concentration. We can use that flow of charged particles to actually go ahead and serve as an energy source which will allow us to convert ADP and free phosphate and convert it into ATP. As long as this flow continues, we get more and more ATP. But you can probably see what's going to happen. Eventually, we're going to have hydrogens build up on this side of the membrane. And eventually, when these things reach equilibrium, the flow will stop. When we get to that point, when we have a buildup of hydrogen on this side of the membrane, we're no longer going to be able to generate ATP. Things aren't going to go from high concentration to low concentration because there isn't a low concentration anymore. If, however, we pump in oxygen to this side of the membrane, the oxygen can combine with the hydrogen to form water. And we can remove that as a waste. This ensures that on this side of the membrane, we're always going to have a lower concentration of hydrogen because it's going to be continually removed by the oxygen. And this flow of hydrogen ions will continue, and we can continue to make more ATP. How much ATP? Hey, from 10 NADs and 2 FADs, we can get up to 28 molecules of ATP for as long as that flow continues. So it really isn't magic. There are reasons for it. All right, if you're a true master of energy reactions, you want to be able to go ahead and explain what happens in electron transport. So sometimes this is easiest if it's done with the diagram. Sometimes you can do it just by paragraph form once again, or if you want to go ahead and sit down with your instructor and just try and practice explaining it, it's probably the best way to do it. If you have questions or concerns, if any of this doesn't make sense, make sure that you ask one of your teachers so that they can help you. Which brings us back to this practice quiz. So, hopefully, if you were listening carefully or following along carefully, a lot of this makes sense. If for some reason you have to go ahead and go back and look at the answers during the video and check your answers, that's fine. It's a practice quiz. But you can be sure that the real quiz is coming, and the real quiz, you won't be able to look any of this stuff up. So make sure you get this stuff down before you're ready for the real quiz. Best of luck. See you in the next video.